Hey, my name is Davila,
I'm Kedar Kirtane from Moffitt Cancer Center.
and we were the co-chairs of a session focused on solid tumor cellular therapy here at CAR-T, and really excited to talk about some of our presentations and I think some of the implications of the work that we presented. My name is Davila. I'm from the Roswell Park Comprehensive Cancer Center in Buffalo, New York.
I'm gonna let Dr. Welty go.
Yeah, I'm Nathan Welty. I'm from the University of Pennsylvania. Eric Smith, Dana-Farber Cancer Institute.
Sebastian Kobold, LMU in Munich.
I'm Yihan Zuo. I come from Roswell Park Cancer Center.
Great. So I'm going to give a very quick summary of my presentation. So what I just discussed was a MUC16 IL-18 CAR T cell that has been developed in collaboration with other investigators at Roswell Park, as well as a AI protein or generative biology protein company. We discussed the development of a novel MUC16 binder that paired to IL-18 and demonstrated its in vitro and in vivo efficacy. We have recently applied to the FDA and was awarded an IND to develop this into a phase 1 trial for patients with a relapsed refractory epithelial or ovarian cancer. We plan to open this trial at Roswell Park over the summer after we complete our clinical trial development. Nathan?
Yeah. So I'm Nathan from UPenn, and my work has been developing novel engineering strategies for myeloid cells. And what I presented today was a new engineering strategy to engineer hematopoietic stem cells for a more durable myeloid cell that could target solid tumors, and showed enhanced efficacy when these cells are given together with CAR-T cells in the solid tumor microenvironment, and the way that these cells can modify the tumor microenvironment to enable enhanced T cell functions. And I think this was really important preclinical work to demonstrate the cooperation of myeloid cells and immune responses, and the fact that we can potentially engineer them for future therapies, and hopefully lay the groundwork for more things to come in the future with new engineering perspectives around myeloid cells.
Yeah, my name is Eric Smith from Dana-Farber Cancer Institute. I shared work that was led by Cedric Louvet in our group, also looking at engineering the hematopoietic stem cell in such a way where the chimeric antigen receptor only is expressed after that HSC differentiates into the immune effector cell type of choice. In our case, we focused on NK cells, and our strategy was also unique in that we engineered the regulatory elements of a lentivirus so that the HSCs can be transduced, but again, that the CAR doesn't express until after the daughter cells have become NK cells. And we confirm the specificity of this approach after screening a large library of potential regulatory element combinations, and also the potency. This is really a long-lived therapy, and we think a safe one that could be transformative to really increase the durability of responses for both blood cancers, but also solid tumors.
So my name is Sebastian Kobold. I'm from the LMU in Munich, Germany. My lab is interested in generating add-ons to cellular therapies to make them work in solid tumors specifically. And today I introduced a strategy where we engineer cytokine receptors to turn suppression into stimulation, and also how we could actually transform heterodimeric signaling into homodimeric signaling to provide a broad range of different effector signals to T cells to transform their phenotype. And by doing so, we would endure their activity over prolonged periods of times against chosen cancer cells for as long as the cytokines supporting that activity would be present. And we could demonstrate the principle of this strategy both in vitro and in vivo work in a range of models. And we believe that this could become a transformative way of enhancing different cell products in pancreatic ductal adenocarcinoma, breast cancer, and other diseases. We're looking forward to translate this work.
Yihan?
Hello, I'm Yihan Zuo, and I come from Roswell Park Comprehensive Center. And our study is focused on next generation CAR-T cell therapy for solid tumor using arming strategy. And our study demonstrates that how IL-36 gamma armored CAR-T cells to reprogram neutrophils and to induce endogenous anti-tumor immunity. And our study indicate that we use an arming strategy which can further enhance CAR-T cell efficacy in solid tumors in the absence of lymphodepletion. And also it demonstrates that IL-36 gamma armor CAR-T cells can activate cancer immunity cycle and inducing the endogenous antigen-specific T cells with the long-lasting anti-tumor efficacy. And this is achieved by mobilizing neutrophils with antigen-presenting cells and also the tumor killing effects. Yeah.
Great. So I really enjoyed this session. I think that while they're all very different talks, they were organized around one theme. How do we overcome the barriers that are present within solid tumor malignancies? And the strategy involved manipulating cytokines, either providing the cytokines through the CAR-T cells or reversing the signal, a negative signal that's provided from cytokines, or novel kind of production systems. So I want to kind of hit on some of those topics for a deeper kind of discussion. So Kedar, do you want to start?
Yeah, and maybe this is not the right forum, but it's still a good question, I think, is, you guys all describe really fascinating scientific technologies to overcome these barriers. Can you comment on the potential scalability as manufacturing complexity increases? As you know, we've had some issues with manufacturing for certain FDA-approved products here. I'm curious whether you've thought about that long term.
So probably relevant for Nathan and Eric.
Yeah. So I can talk about that a little bit. I mean, we've had a lot of experience at our center with engineered HSC products using CRISPR editing technology, which is what I use to generate my cells. And we, I think from the get-go, have been very intentional about trying to use the same sorts of GCSF mobilized normal donor cells that we would use if we were to translate this into an, a regular system. So we've tried to maximize translatability from the get-go. I think there are a lot of unanswered questions in the field about number, quality, et cetera, in the HSC, engineered HSC space in particular. And there are going to be a lot of probably regulatory issues around that when you're engineering these cells, either with viruses or with CRISPR-based technologies that we have to anticipate. I mean, I think, we can learn from what we've done with CAR-T cells, but I think there's going to be some unique factors that are specific to HSCs, and those parameters, I think, clinically will have to be developed kind of as we think about getting these closer to translation.
Yeah, I can briefly add to that, and this is a super exciting time to be doing gene and cell therapies, as you know, and there are now FDA-approved HSC modified cells, not in cancer immunotherapy, but for sickle cell and hemoglobinopathies, and so we really want to leverage that experience. We think for our cancer patients to be mobilized and collect HSCs, if this was done ex vivo, is certainly possible. We do that for auto transplants. We think the numbers of cells that are needed are reasonable to obtain. But ultimately, I think the goal of going direct in vivo and mobilizing HSCs, targeting HSCs, while that I think there still is some development to be done clinically is certainly feasible with the tools that we have today.
Great. So now I wanted to kind of talk a little bit about the cytokine talks. So I present on IL-18, Yihan, IL-36 gamma, Sebastian on some kind of features of cytokine receptors in general. So there's obviously lots of great cytokines that have impacts on the immune system, T cells, the microenvironment, IL-2, IL-7, IL-12, 15, 36, 18. What are kind of the defining features that we should look at as designers of CARs to go, these are really the important features to be able to combine with a CAR T cell, specifically when attacking a solid tumor cell therapy? So I'll kind of pitch that to you all. Sebastian?
Yeah, sure. So, I mean, to me, at least, or to us, I think a key feature is dose response. Because I think which was also the reason why we chose to pivot more to the receptors than to the cytokines, which we do too, simply for the reason that although I think the topic of being able to reprogram the TME is very compelling, I mean, I'm not sure that we really understand how much is enough and how much is too much. And in that sense, it makes it really difficult to say, okay, is it IL-18? I mean, there's great data already out there for IL-18, or is it maybe another one that would be better? And if so, and when should we stop doing that? So I think that's a little bit of the challenge. Whereas if you think of just of a cell autonomous system where the thing that's being fed is just the T cell, that becomes maybe a bit more controllable in terms of understanding how much the tumor already has and is producing constantly as a function of the cells that are there. But I think the question is open, and obviously in your data it looks different too, right?
Yeah, and in terms of the translational study, actually the cytokine armored CAR-T cells, the function of them are immunomodulatory functions beyond killing ability, because well-known the lymphodepletion is very important step for CAR-T cell preparation. But in our study, we found that the lymphodepletion also can disrupt the interaction between the immunomodulatory effect with the cytokine armored CAR-T cells. So whether the role of lymphodepletion is important or not, this is need to be investigated. And the beauty of armored CAR-T cells, they rely on the endogenous T cell response rather than the CAR-T cell persistency. Yeah.
Right. Well, I love the fact when you kind of highlighted translation of some of these ideas in cell therapies, because ultimately that's probably the most important thing that has to happen with anything that we do. We have to put this into patients and find out, what are the toxicities, what are the potential benefits? Can we improve on this in an iterative fashion? There's already been publications of clinical trials using IL-18. So that information, I think, is very valuable that we can kind of go, are these things that we can kind of build on? Another topic I wanted to talk about, we didn't necessarily present this, some adjacent stuff, but I almost feel like if there's a cell and gene therapy discussion, always have to include in vivo gene therapy. That is kind of hot. Industry's moving towards that. Academic kind of labs are that. I want to kind of hear just people's just general ideas involving in vivo gene therapy. Is this something that is ready for prime time? Is this really going to replace adoptive cell therapy? What are people's thoughts on this? Because partly, I just want to know for myself so I can steal good ideas. Throw in whoever. Sebastian?
Yeah, I mean, I hope we can all agree that, if that would work, I mean, that would be a game changer in everything we do because of scaling, because of cost, etc. To me, one of the biggest questions that still needs to be answered is, to which level do we manage to get such a strategy that helps us to get away from the need from non-myeloablative or chemotherapy or preconditioning in other sense, right? Because I think it's very clear in all the autologous and allogenic studies that we've seen that you need that. And everyone that had cohorts without did not see activity and then closed the cohort and then went on to the one with this. And to me, that's in patients. Now I'm not sure about that. I mean, there've been these few patients, myeloma patients that look good, but short follow-up. So I don't know if especially in solid tumors, to which level you can get away without that.
Yeah.
I mean, I think also when it comes to in vivo, I think a big challenge that the field is going to have to address is cell number. When you manufacture an ex vivo product, you're giving, a defined cell number, typically in our clinical trials, and in our, there are manufacturing release criteria for all of the commercial cell therapy products, for instance. You probably don't need as many T cells as we're giving people, but how do you quantify that when you're giving this in an in vivo manufacturing system? And I think that becomes even a bigger question when you're moving outside of T cells, because as I talked about, the CAR is going to expand your T cell population naturally in vivo, but that doesn't happen with some of the other immune cell types that we're talking about engineering in vivo. So then you have to think about, well, is there an expansion strategy for those cells? Or is there a selectable marker that we can induce? And one of the things that I didn't talk about with my strategy is that by knocking out and knocking in at the same time, now we've introduced a naturally selectable marker, because only our knocked out, knocked in cells will be knocked out for the gene. So you can give an anti-CD33 antibody, for instance, deplete all of the CD33 positive cells and enrich for the engineered cells that we're trying to do. And so I think employing strategies like that for other cell types might become important as well when we think about moving to an in vivo setting.
Yeah, I can
Eric,
just add.
I want to ask you though as well, because your system is very adaptable to in vivo, but it requires lentivirus, because you can't really do with RNA because you wouldn't get that persistent gene engineering. So But RNA is attractive as well from a safety kind of perspective. So how do you kind of think of lentivirus versus RNA for in vivo?
Yeah, I can just preface that by some of the field in general. And I'm been very impressed with the data we only have, with all the caveats, a few patients, a few months of follow-up. But the fact that we're seeing deep responses in nearly all these patients without lymphodepletion, I think is very encouraging. And then to your question, lentivirus versus RNA, I think there'll be different indications for both of those. RNA, of course, you're not getting the persistence, but you have added safety that may work really well if there is a way to target maybe minimal residual disease or certainly autoimmunity, or requiring repeat dosing similar to a T-cell engager. Whereas the lentivirus approach, either certainly through targeting HSCs, as we're doing, is still a one-time treatment where you're going to have very long-term production of these modified immune effector cells, and even with T cells, if there's massive expansion, could potentially overcome that.
Fantastic. So at this point, great discussion. I think that's the great kind of aspect of this meeting, is being able to talk with our colleagues about a great research in things that have implications within the field of cell and gene therapy. But I'd like to close out this session now, and on behalf of my co-chair, Kedar Kirtane, and my co-panelists, want to thank everyone for taking their time and listening to this discussion.